welcome everybody for this genomics track session. With much further ado, will introduce the first speaker as we don't have much time for each of them. So the first speaker is Jack Lanchantin from the University of Virginia. So whenever you want, Jack.
Sure, thanks. Okay. Hi, everyone. So I'm going to be talking today about our work called Graph Convolutional Networks for Epigenetic State Prediction using both sequence and 3D genome data. So this is joint work with my advisor, Dr. Yanjun Chi from the University of Virginia. So just a quick preliminary note before I get going. So we use the following words interchangeably: epigenetic state with chromatin profile, and 3D genome with chromosome conformation. So throughout the talk, I'll sort of use those interchangeably. So alongside genes are chromatin elements such as transcription factor binding and histone modifications, represented by these colored shapes above the DNA here. And these are regulatory factors that control whether a gene is expressed or not, making them important to predict and understand. So we define the chromatin profile or the epigenetic state of a DNA window Xi as the chromatin elements that are present at that location. So this includes transcription factors, histone modifications, and accessibility. So epigenetic state prediction is then the process of taking DNA sequence windows as input and outputting the active chromatin elements at those windows using some model F. So given some new DNA windows, the model hopefully will be able to accurately predict the chromatin elements at each of those windows. So previous state-of-the-art methods for this task predict the state of each DNA window independently of all other windows. So we'll take one window, predict the chromatin elements that are present there, move to the next window, predict the chromatin elements, and so on. So these types of models are fairly accurate due to the influence of local sequence features on the chromatin elements. So for example, maybe this red diamond shape might be representative of a transcription factor, and that might bind to window X2 because of this subsequence TATGA. However, some elements such as this blue circle, which might be representative of a different transcription factor, may be present with no sequence features in the window. So one hypothesis for this is due to what's known as chromosome conformation, or the 3D genome, where DNA actually folds in the chromosome. And because of this, windows may be influenced by sequential features which are really far away in the linear space, but really close in the 3D space. So our goal is to incorporate this type of structure into our prediction method so that we can consider these long-range interactions for predicting these chromatin profiles of each DNA window. So this 3D shape of DNA can be represented by high C data, which indicates physical contact between two windows in the genome. And these contacts can be represented as a graph where nodes indicate DNA windows and edges indicate contact between two windows. So this type of 3D structure of the genome can really just be represented by an adjacency matrix that we call A, where again, the nodes are the DNA windows, and a positive cell would indicate that there's some sort of contact between those two windows, which is representative of the 3D shape of the genome. So our proposed method to incorporate this data we call ChromeGCN, which is the first deep learning method to combine both sequence and 3D genome data for chromatin profile prediction. So it's about incorporating this type of 3D structure via high C data into this prediction method to predict things such as transcription factors, histone modifications, and so on. So formally, our goal is, given DNA window Xi, we want to predict the probability of a set of epigenetic labels that we call Y hat sub I, which is just a vector of L total chromatin profile labels. And our method, ChromeGCN, consists of 3 different modules, which I'll go over individually here. So the first module is extracting local sequence features using a convolutional neural network. So this module FCNN maps each local sequence window xi to a vector that we call hi of dimension d using a shared convolutional neural network. So this is done for each window independently of one another, which it takes in, for each window, it takes in the sequence and outputs a vector representation that encodes that sequence information. So once we have these vectors H, the second module, which is the main contribution of our work, encodes this 3D conformational relationships between all windows, between all window vectors using a graph neural network. So without going into the details of how this graph neural network works, each of these HI vectors, which represent the sequence features for a particular window, is updated to a new representation that we call ZI using a function of itself and its neighbors indicated by the high C adjacency matrix A. So now this output vector ZI for each window encodes both the sequence information and neighboring sequence information that we're using from the high C data. So once we have these Z vectors for each window, the final module just is simply a linear classifier to predict the L epigenetic labels for each of those windows. So sort of putting all this together, our model looks like this, where the first step is extracting local features from the sequence using a convolutional network. The second step is extracting long-range 3D interactions using graph neural networks. And finally, the prediction module to predict the epigenetic labels for each window. So I'll just quickly go over how we try to validate this model. For epigenetic state labels, we use all transcription factor, histone modification, and DNA accessibility data from ENCODE using two commonly used cell lines, GM12878 and K562. And for high C data, we use the interchromosomal contact matrices, which tells us edges within a specific chromosome. So based on this high C type of data that we're using, we split the data by chromosomes using 16 of them as training, 3 as validation, and 3 as testing. So each sample is really one chromosome where we want to predict the labels for all windows within that chromosome. So if you want to use this model to predict on new windows, you really need a full chromosome because we're using all the interactions within that chromosome to predict windows. So we compared 2 commonly used independent window-based deep learning methods, the first 2 here, and we introduced 2 variants of our method, the proposed Chrome GCN model that I talked about, as well as a recurrent neural network version, which really just replaces the graph module with an RNN. So it uses the full linear chain of windows rather than this known 3D structure. So it's just a way to validate whether using the known 3D structure is important, or if we want to just consider all the linear chain of windows. So we find that across both cell lines, incorporating this long-range structure into the prediction model helps improve accuracy when compared to these independent window-based methods. But most importantly, comparing the GCN approach versus the RNN approach, we found that using these known 3D relationships or the known conformational structure of DNA helps the best out of all the models that we tested. So lastly, I'll just talk about a post-processing method that we call high C saliency map. So this is after we've trained the model and want to analyze what the model learned. So while high C maps tell us which pairs of DNA come in contact with each other, they don't tell us which contacts are important for a specific chromatin element to be present. So in other words, it just tells us where the contacts occur, but it doesn't tell us which chromatin elements are affected by those contacts. So we propose what we call high C saliency maps, which is just a method to identify the important 3D relationship of the important contacts for Chrome GCN's predictions. So this is done by taking the gradient of the output y hat for label L with respect to the high C adjacency matrix A. So this will tell you the important edges in this high C graph for a particular label L. So just to show a quick example of what I mean by that, here's an example of our high C saliency map for the transcription factor YY1 in chromosome 8 of cell line GM12878. So what this shows here is the importance of all 500,000 edges that we use for this chromosome, where the darker the line is, the more important that edge was for predicting the ground truth YY1 value. So we can kind of zoom in on particular subspace of this full adjacency matrix and see which windows are actually important for this YY1 transcription factor. So just in conclusion, we propose a new method for incorporating chromatin contact information for this chromatin profile prediction task. And we found that incorporating this type of 3D data helps improve prediction accuracy. And lastly, we propose a new method to find the important high C edges from the high C graph. So our code and data, all the data that we use in the code is available on GitHub. And I just want to thank my advisor, Dr. Chi, and my many collaborators over the past few years.
So thank you, Jack. So I'm checking now the questions. We have a couple of questions. We have one from Peter, who's asking, is the window size fixed? Can windows overlap?
Yeah, so the window size is fixed for our training. We use windows of 1,000 length, and they're non-overlapping, but we can adjust that. I mean, that was just the formulation we used. We actually tried to use the same data processing as the deep C and expecto methods. So, but that it's just an arbitrary choice, and that can actually be changed.
Okay, thank you. We have another one by Davide Bau. What was the resolution of the high C data that you use in your models?
Yeah, so we try to use the lowest resolution possible, actually. So we use 1,000 length windows, and that was partly chosen because the lowest resolution for for one of those cell lines that we used was 1,000. So we tried to make the window size the same size as the resolution from the high C data. If the high C resolution, the lowest resolution is higher than that, then we can just we can change the window size for the sequence information. So it's really, it's kind of dependent on how low the resolution is for the high C data, but that can be changed arbitrarily.
Okay, thanks. So we have time for the last question by Jens. He's asking basically about the overtraining, like you have few genomes to train the network. How do you avoid the overtraining problem?
Yeah, so that's a good question. I mean, you know, I think ideally you would want to train. I think that this type of model is more for the analysis of a particular genome. You do run the risk of overfitting it. We tried to train on a specific subset of the chromosomes and test on a held out set of chromosomes. But yeah, I mean, it would be hard to transfer this model between different cell lines because each new cell line has a different high C data, but yeah, I think that is an issue, but we tried to at least account for that in our training testing splits.
Okay, thank you, Jack. So
thank you.
We have no more time for questions. We should move on to the next speaker, who is Huy Nguyen from Iowa State University. So Huy, whenever you want, you can start.
All right. Hi, everyone. Can you guys see my screen?
Yes.
All right. So my name is Huy Nguyen, and I'm from the Department of Computer Science and College of Veterinary Medicine from Iowa State University. So today I want to present my paper, which is about finding autologous gene blocking bacteria. So the motivation for the study is to look into the evolution of complex system, and in bacteria, gene blocks can be considered a complex system. So the problem for this talk is how we identify the autologous gene block in bacteria using an event-based method. So this talk mainly evolved about how we show the problem actually really hard, like by showing it's pretty hard. And then because it's hard, so we show a greedy algorithm that is a variant of the set cover greedy algorithm that can still give us an approximation result. And then I'll discuss some of the future work. So gene blocks are genes that are collocated, usually sharing the same pathway or protein complex. In the genome, we say two genes are collocated if the distance is less or equal to 500 nucleotide. The operon is a special case of gene block where the cluster of genes are co-transcribed together by polycistronic mRNA under common regulation. So previously in a paper, we have described three main events for evolution of gene block. So let's say in our reference gene block or operon, you have gene 1, gene 2, gene 3, gene 4. And in the target genome, you find ortholog 1, ortholog 2, ortholog 3, ortholog 4. And then if there's a distance more than 500 nucleotide between gene 2 and gene 3, then we say there's split event. If we cannot find ortholog of gene 4, then there's a deletion event. If we find a copy of ortholog of gene 4, we say there's duplication event. So the relevant gene block problem can be visualized as follows. Given the reference operon A, B, C, D, and E, and in the target genome, you have several gene blocks such as AB, CD, or ADE, and BC. Then there's several potential or candidate orthologous gene block, which could be like either AB with CD or CD, ADE, etc. So basically all the subset available from the target gene block. The main problem is how to find a minimum relevant gene block so that it minimizes three distance that are discussed above toward our reference operon. So to formalize this problem, we're giving a reference gene block, which is R, in a cluster of homologous genes in the target genome. Then we want to output O prime, which is a subset of O, so that it minimizes the sum of the three distances. And in this talk, I'll just simplify our problem by not consider the duplication distance. So we only have the deletion distance and the split distance between O prime and R. So using some mathematical transformation, we end up with a simplified version of a set of genes of the reference operon, which is set S, and a cluster of homolog in the target genome, which is set C. Then one output C prime, which is a subset of C. So it minimizes the following function. This function basically the summation of the size of C prime and the number of elements in S that was not covered in C prime. So if you're familiar with a set cover problem in computer science, so this is pretty similar. So the set cover problem, just to state it, is a problem with giving a set of element or universe and collection of subset of S. Then we want to output C prime so that C prime cover S. And also the size of C prime is minimized. So we'll show that a problem is anti-hard by reduce it to the set cover. So with a very typical way of doing that is giving an instant SC of the set cover problem. I construct the same instant SC of the relevant gene problem. I prove two things. So the left-hand side is giving a solution of the relevant gene block. I can construct a solution for the set cover problem. And given a solution of the set cover, I can construct a solution of the relevant gene block. So for the left-hand side direction, we have the first claim is that if C prime is a solution to the relevant gene block, then C prime by itself would be the minimum set cover of the union of C prime. So for example, given a reference operon ABCD, E. Then you have a cluster of gene blocks that come from the target genome, which is the EADE, etc. Then we can consider C prime to be some arbitrary result of the solution of the relevant gene block problem. And in this case, it contains of gene block A, B, C, D. Then we will show this is also the minimum set cover of the set A, which contains gene A, B, C, D. So to prove it, we assume there exists another set C1, so that C1 actually can cover A, but the size of C1 is less than C prime. Then if you recall the function of d of S of C1, when we plug in C1, then it's just the size of S minus the union of C1 plus the size of C1. The size of C1 now actually is less than the size of C prime, as we assume right here. And then we know that C1 cover A, so therefore union of C1 basically is union C prime. Therefore, distance of S to C1 is actually less than the distance of S to C prime, which contradicts to C prime is a solution to relevant G block. So therefore, claim 1 whole. And then the second claim is basically saying for every element that was not covered in C prime, then what the element C can contribute at most is one more gene to the set C prime. And C prime is the solution to the relevant G block. So to prove this, we assume that exists another element, let's say C, that can contribute at least 2 to a set C prime. We'll show that basically our C prime should have included this inside of it by default. Then to prove it, we assume that there exists one. Then we consider C1 to be the set of C prime union of C. Then we can, the union of C1 now basically is the union of C prime minus plus whatever it can contribute. And then as we say it's at least 2, so the minus will give us less or equal to minus 2. And the size of C1 now is whatever the size of C prime plus 1, because C prime and C doesn't intersect. So therefore you have C prime plus 1. So negative 2 give you basically just the distance from S to C prime minus 1, which is less than itself. So therefore this contradicts to the assumption again. And then claim number 3 is for every element that can give one more gene to C prime, then we can always construct C1 as the C prime union with C, so it still be the minimum set cover. So therefore, using those 3 claims, we can derive a set cover as from C prime that has the minimum cost and in polynomial time. Then therefore, we can construct a solution from set cover problem using a solution from the relevant G block problem. Now for the right-hand side direction, consider C prime as solution for the set cover problem. Then we plug in C prime for the distance function. Then the psi of S minus union of C prime basically 0 because cover everything. Then what the distance of S to C prime, just the psi of C prime, right? So now let's assume if C prime is not a solution to the relevant G block, then we have again a set C1 so that the d of S to C1 is less than the distance of S to C prime. Then using the same construction as the show above, we can reconstruct a set cover using this as C1, so it still have the cost of the same d of S of C1. So therefore, this new set, we have the size less than our size of C prime, but still cover everything, which contradict to C prime as a solution to the set cover problem. So therefore, given a solution set cover problem, we can construct a solution to the relevant G block problem. And with those two left-hand side, right-hand side reduction, we have shown that our relevant G block is at least as hard as to the set cover problem. So because it is really hard, then it's unlikely to be able to solve it exactly in polynomial time. Then we provide a greedy algorithm to solve it. This one is a variant of the set cover problem, and where we know that the set of the orthologous gene block might not cover the whole gene set. So therefore, we initiate S prime as a union of C, then C prime is empty. Why our S prime is not empty? That we keep finding the element that maximize, or basically mean it covered most from S prime. Then we remove it from S prime, and we include this new element C into our C prime, then we return C prime. it actually share the same upper bound with the normal greedy algorithm, log natural n, although we have this step right here. So the proof is actually not trivial. You can find more in the manuscript of finding authoritative gene blocking bacteria, so I won't elaborate it here. So regarding the result, our method actually is pretty fast. And it's faster than the previous method, and which can be found also in the paper. So here I will just tell you about what we have done. So basically, we have formalized the problem of finding authoritative gene blocking bacteria. We also proved that the relevant gene block problem is NP-hard, and our 3D method, the approximate, actually has a better running time and optimality than the previous method. So for future work, because we are only we don't take into account of duplication cost right now, so we would want to include that. So definitely right now the simplified version is already NP-hard. So if we input like the duplication, it will still NP-hard. But what matter is, does our greedy algorithm still give the upper bound of the solution? Then also we haven't taken account of gene order because we consider the block as a set of genes or multi-set of genes. So we lose that gene order information. So it will be interesting to take into account of those. And also we have not considered horizontal gene transfer event. We only care about vertical transfer event right now. So finally, I want to thank Professor Ido Freebird, Professor Oliver Ornstein, and Matthew Markin for this for helping me with the manuscript, and Ido Freebird for guiding me through my PhD career, and Oliver Ornstein for helping me with the CS component. I want to thank Ido Freebird Lab for helping me with the study. And this is the code that I use in the paper, and this is the paper title. I also want to thank NSF for funding my study at Iowa State University. So now I'm open to questions. So let me stop sharing. All right.
So thank you, Huy. I don't see any question in the panel, so I will ask one or a couple myself. So because this topic is of interest to me, so will your algorithm consider the possibility of co-orthology? Like you have the same block is simply duplicated in another genome, but the whole block, not a single gene. So you have twice the same block and the same relationships between the gene order. In that, what would you do in that case?
That is very interesting question. So definitely, sometimes we have a duplicated of the whole block. But the thing is, right now, like it doesn't happen too much of the time, and also what we consider right now is our definition of authoritative gene block is a set of gene block, and gene block can be multi-set. So we allow duplication within a block, but not duplication of the whole block. So if there's a duplication, then we just consider as just one block, like all of them just one block of genes.
Okay.
And then yeah, so it's a genes, it's a set of blocks.
Okay, it's clear. And also you said that you consider you don't consider the gene order within the block. So
No,
I assume you also not considering, you know, the strand direction of the gene, no? Like if one gene is simply flipped around but kept in the same. So this is also not considered.
Actually, if it is split, it is reversed when transcribe stuff like that. Then we actually consider it a split operation.
Okay.
Yeah. So that's a split operation right there. So that's how we consider that. Yeah.
Okay. So let me check if there's any new question in the panel. I don't see any, but we have to move anyway to the next speaker. So thank you again.
Thank you.
Okay, and now our next speaker is Sayaka Miura from Temple University. So Sayaka, when you want, the floor is yours.
Okay, thank you. Metastasis is the spread of abnormal cells between tumor sites. For example, when a cancer cell in skin melanoma moves to brain, it's called brain metastasis. Most of the cancer death is due to metastasis. Over time, many mutations accumulate, producing genetic heterogeneity. Therefore, we can infer evolutionary relationship of cancer cells. This is a phylogeny of cancer cells. Each tip of this phylogeny is cancer cell. And there are many cancer cells with identical genotypes. We call them clone. So in this phylogeny, there are C1 to C5 clone. These clones are found in liver or colon. By using this information, we can infer migration paths. There is a method called machina which can infer migration paths by using phylogeny of clones. In this method, first we estimate the ancestral location of clones. In this case, ancestral clones are A1 to A4. So machina is a parsimony approach. By using parsimonious criteria, it predicts these ancestral tumor site. In this case, it predicted all of ancestral tumor site is colon. Therefore, we obtain colon to liver migration path. However, this method doesn't use information of branch length. I will illustrate the importance of branch length by using this simple phylogeny. This is a phylogeny of 3 clones. All of the clones are found in 3 different tumor sites. Without branch length information, we cannot determine the ancestral tumor location of this A2 clone. As a result, we will obtain 3 equally parsimonious solutions. This is the case when we have the information of branch length. In this case, ancestral clone A2 is connected to clone C2 with very short branch length, and C2 clone is found in M1. So we can think this A2 clone is also found in M1. Then we can find a single solution like this. We developed a new method called Pathfinder. Pathfinder uses clone phylogeny and branch length. I will explain Pathfinder approach by using this simple phylogeny. This is a phylogeny of 3 clones, C1 to C3. Each clone is found in P, M1, and M2. This observed tumor site is expressed as X. The goal is to predict the ancestral tumor site of A1 and A2. This unknown ancestral tumor site is expressed as Y. B is branch length. So we will compute posterior probability of each ancestral clone in each possible tumor site. For example, we can compute posterior probability of ancestral A2 clone found in tumor site M1 like this. We similarly compute posterior probability of A2 found in P and M2, and then select a tumor site which receives the highest posterior probability to assign the tumor site of this ancestral clone A2. We repeat this procedure for other ancestral clones. In this case, another ancestral clone is A1. In this case, we find P and M1 received the highest posterior probability for these ancestral clones. So we will put these tumor assignment like this. We predict migration event when 2 nodes have 2 different tumor assignment. So we predict P to M1 and M1 to M2 migration events. At the end, we summarize all of these predicted migration events like this. We tested the performance of Pathfinder by using simulated datasets. Our simulated datasets contain 6 to 30 clones, 10 to 100 SNPs, 5 to 10 tumors. We generated 4 different seeding types. In the first type, there is only a single migration event between two tumor sites. In this case, some tumor sites receive two clones from another tumor site. In this case, some tumor sites receive two clones from two different tumor sites. Lastly, in this case, some clones in metastatic tumor site move back to primary tumor. First, we examine the results of dataset with this shading type and find that Pathfinder performed very well. For most of the dataset, Pathfinder produced the perfect results. Next, we examine dataset when Pathfinder didn't produce the perfect results and find that those datasets were impossible. This is an example. This is a simulated clone phylogeny. To generate our dataset, we sampled clones from this simulated dataset. In our dataset, we didn't sample this C20 clone. So in our dataset, there is no information about M1 clone. Therefore, it's impossible to infer migration event about the M1 tumor site. These results indicate that it's very important to have a large number of clones from each tumor site to obtain accurate migration events. Similarly, Pathfinder performed very well for the dataset with this shading type. Again, we examine dataset when Pathfinder didn't produce the perfect solution. Here is another example. In this case, ancestor clone A2 is connected to C14, C42 and C24 clones, which are found in two different tumor sites. These clones have similar branch lengths leading to A2 clone. Therefore, in this case, it's impossible to determine the ancestor tumor location of this clone A2. So Pathfinder produced two solutions. These results indicate that it's very important to have a large number of SNPs to find accurate inference. Similarly, Pathfinder performed very well on the other datasets with these shading types. And we found that Pathfinder didn't produce a perfect results only when these datasets were impossible due to insufficient sampling or similar branch lengths. Next, we computed F1 score to further evaluate the performance of Pathfinder. For each inferred migration plot, we counted the number of paths inferred correctly, the number of paths not found, the number of incorrect paths. And then we calculated precision and recall. F1 score is the average of these two values. For each inferred migration plot, we computed F1 score, and then we calculated the average F1 score for each shading type. We found that Pathfinder produced the highest F1 score for this dataset, and the F1 score declined when the dataset contained more complex migration events. We next compared these results with Machina. So we computed F1 score for each Machina results. Here is the results. In general, Pathfinder produced better results than Machina, especially for these two shading type. Pathfinder produced much better F1 score than Machina. Since Machina doesn't use branch lengths, this results indicate that the use of branch lengths is very important to produce more accurate results. Lastly, we tested the performance of Pathfinder by using actual empirical dataset. We obtained this dataset from Hoadley et al. Paper. This is clone phylogeny reported in this original study. Circles are the clones, and colors represent tumor site. This is breast cancer, so primary tumor site is breast, which is indicated with green. And this dataset has 5 metastatic tumors. Pathfinder produced this migration path. So lung and brain were shielded from the primary breast tumor, and Pathfinder predicted many migration events between metastatic tumor sites like this. We found these migration events were reasonable comparing to the clone phylogeny. This is the clone phylogeny. The white circle is predicted ancestral tumor site by Pathfinder. We found all of these inference is reasonable. For example, Pathfinder predicted ancestral clone A2 was in lung. This clone A2 is connected to clone C2 with very short branch lengths, and C2 clone is found in lung, so we can consider ancestral clone A2 was also in lung. Similarly, A4 and A5 clones were predicted to be liver, and many clones in this section of phylogeny were found in liver, so we can think these ancestral clones were also found in liver. From this prediction, we can find liver to kidney metastasis. This is shown here in this plot. This result was very different from the original study, where they proposed all metastatic tumors were shaded from the primary tumor. However, this proposal was not supported by the clone phylogeny. Therefore, Pathfinder can produce more reliable inference on actual empirical data analysis. In conclusion, method to infer migration events is very important to answer fundamental questions. For example, how often primary to metastatic tumor migration events happen. We developed a new Bayesian method called Pathfinder. Pathfinder uses clone phylogeny and branch lengths. We showed that Pathfinder performed very well. We also found that it's very important to have a large number of clones and a large number of SNPs to obtain reliable inference. Thank you. If you have any questions, I will take it now.
Okay, thank you so much. We have some questions. No, we have no question. Oh, yes, one question, sorry. From Sophie. Do you plan to include the uncertainty of the phylogeny in your inference?
So, sorry, could you repeat it?
The question is whether you plan, I mean, sometimes the trees have uncertainty on the topology, like the bootstrap value or, you know, how much you rely each branch. So are you planning to include these values on your inferences?
Currently, Pathfinder cannot take that kind of supporting value, but now we are trying to incorporate that kind of values to deal with some uncertainty in the clone phylogeny.
Okay, thanks. And so I have one question myself. So all the phylogenies you show were rooted.
Yes.
So my question is, how do you root the tree? You need to include, like, you know, the normal tissue or something to have it rooted?
Yes, so we use normal tissue. The phylogeny we showed contain only somatic mutation, so we can compare tumor variants and variants from normal sample, and then we can root the tree.
Okay, thank you. So I don't see any other question. So we move to the next speaker. So thank you again, Sayaka. So our next speaker is Sarah Christensen. She's coming from, well, she's connecting from the University of Illinois. So whenever you want, Sarah.
Great. Thank you, everybody, for joining me today virtually. I'm excited to be here. So today I'm going to be presenting work on detecting evolutionary patterns of cancer in cancer patient cohorts using consensus trees. And so this is joint work along with other professors and students at the University of Illinois, as well as Nicholas Chia at the Mayo Clinic. So our last presentation actually led us into this quite well. So as we just saw in the prior presentation, cancer is really an evolutionary process. So we have some initial set of founding mutations, but as the cancer progresses and cells continue to grow and divide, new mutations are introduced. And eventually in mature tumors, we observe heterogeneity in the cells. So different mutations cluster into different cells of the tumor. And as we just saw, this process is typically represented by a phylogenetic tree. And so the nodes of these trees, tumor clones, as well explained previously. And then on the edges of these trees, we see new mutations that are introduced, differentiating the parent clone from the child clone. And so there's many sort of interests in why we might want to be able to understand the different evolutionary history of a particular cancer patient. One, just to better understand evolution in cancer. But I think one of the sort of longer term goals is that if we can really better understand and estimate tumor phylogenies, potentially they can help us improve stratification of cancer patients into subtypes. So maybe this is in different treatment plans or different prognoses, and understanding really like what makes different cancer patients similar. And so the first step in this longer term goal is really to be able to identify repeated patterns across different cancer patients based on their evolutionary histories. And so when we're talking about patterns here in this context, I'm really talking about repeated orderings of recurrent driver mutations across patients. But a kind of a current challenge to being able to find these repeated patterns is that as we go on estimating patient trees kind of independently, there's a lot of uncertainty kind of highlighted in that question that we just got in the different topologies of particular cancer patients' evolutionary history. So here is an example from a recent paper in the New England Journal of Medicine. This particular patient had exactly 17 different alternative hypotheses for how their tumor could have evolved. And so basically kind of some of the current methods only using individuals' patients' data are underspecified, and we have this ambiguity. That ambiguity, one, makes it hard to understand that particular patient's tumor, but then two, helps basically obfuscate these repeated patterns, so it becomes hard to identify what is happening again and again at the cohort level. And so there's been sort of recent interest in solving both of these problems concurrently. The idea being, if you can look at cancer patients at the cohort level, we can, one, help use other patients and the patterns we observed in those patients to help reduce ambiguity in individual patients through the idea of transfer learning. And then also, as we observe kind of what are those things being transferred and learned, we can identify these repeated evolutionary patterns that are getting shared. And so two recent methods I just have here that really pioneered this work are Revolver and Hintra. There's also a recent paper in ISCB 2020 ConNet that was looking into a similar thing. So it's a growing and exciting kind of area that we're looking into. And so one of the kind of current limitations of these methods is that they don't explicitly account for different cancer subtypes. So as we know, for a particular primary tumor location like breast cancer, not all patients are the same. They may have different subtypes. So when we think about learning, sharing patterns, we might want to account for that. There's also difficulty in scaling, especially with Bayesian methods in certain cases. And then we also have trouble dealing with varying mutation sets as well as mutation clusters, which are kind of key components that often come up in real biological data sets. And so in our approach for this paper, we first posed an optimization problem called the multiple choice consensus tree problem. And this is really building on a line of work out of our lab, looking at summarization using consensus trees in this cancer context, as well as a corresponding algorithm recap, which is looking at basically a heuristic for solving this optimization problem. And so the idea at a high level is that our approach is similar to other methods, leveraging common patterns of evolution in patients, but emphasizing that we're finding these patterns in subtypes of patients and using them to resolve ambiguities in the patient data. And so hopefully it'll be a little bit more clear as I go through the specifics. So here's our optimization problem itself. So this is the input is basically a cohort of cancer patients and their evolutionary trees, or the different alternative evolutionary trees that have been proposed. So here we have this set of patients, and each patient may have multiple trees associated with them, as well as a parameter K for the desired number of clusters to output. And so I'll walk through the output of this problem sort of slowly, because it's there's a couple components to it trying to resolve all at once. So at first we want to be able to select a tree from the input trees for each patient that we feel is sort of more supported, that we feel is more likely to be their true evolutionary history. So this is reducing ambiguity in the input set. The next thing we'd also like to do is to be able to assign each patient into a cluster, up to K clusters. And this is really getting the idea of like a cluster intuitively is trying to represent a particular subtype. And then finally, for each one of these subtypes, we'd like to be able to construct a consensus tree, essentially summarizing what are these repeated patterns that we're seeing across trees within this subtype. And so this is, you know, addressing the limitation of when we're learning different patterns across patients, it's really going through this consensus tree that's representing a particular subtype. And so there's a selection and assignment and construction component to the output. And we want to do all three things in such a way to minimize the sum of distances between essentially a patient's tree and the consensus that summarizes it. And so just one thing I kind of wanted to highlight when we think about distances that was kind of interesting when we were looking at this paper, and something I think that can be continued to be explored, is we wanted to be able to account for varying mutation sets. And so in practice, we found that this particular distance works quite well. So in the numerator, we have the symmetric distance between the edge sets of two trees, and this is typically thought of as a parent-child distance, and it's pretty common. We also include the symmetric distance between the mutation sets of the two trees. And then we normalize this basically by an upper bound bounding the numerator, putting the distance between 0 and 1, which is helpful so that certain patient trees that are, you know, much larger don't ultimately dominate our optimization function. And so just to summarize some of our results, we have some theoretical results proving the hardness of this particular MCCT problem. And we also have a gradient descent heuristic we use to be able to address this problem. And we have a subroutine with model selection to help identify a particular K or number of clusters to use in practice on a data set using a kind of sweep. We believe that this helps to address some of the prior limitations, explicitly accounting for patient subtypes, also allowing for different mutation sets and scaling to larger sets of data with many more mutations. And also something I think was kind of neat was that we included a dynamic programming subroutine to help handle mutation clusters. And this potentially could be an approach that helps in other contexts when people are trying to deal with mutation clusters. And then finally, we implemented our algorithm and tested it on both simulated data as well as biological data. So I'll get into some of those results now. So without getting into too much of detail with the simulation procedure, which is all covered in our paper, I really just wanted to highlight that our simulation procedure kind of tried to test, one, when there's no subtypes, sort of all patients are the same, which is really the model under which some of the other methods were developed, as well as evaluating ground truths where there are indeed subtypes. And so we varied the number of subtypes then. And we did this under a number of different parameterizations, and we had over 600 simulated instances in total. So this is some of our results, and here we just combine over all of these simulated instances. But in our supplement, we break this out by different parameterizations, but here it's just combined, and I think it represents kind of the trends we see in general. So here we're looking at the question, is the correct patient tree selected? So this is the reducing ambiguity question. And we see that when there aren't patient subtypes, all the models essentially, or algorithms performs the best. This is when K equals 1, and they do a good job of identifying the correct patient tree. And then the trend that we notice is that though, as the number of subtypes increases, some of the other methods deteriorate in performance or perform less well overall compared to our approach of recap, which is here in orange. And we looked at a number of ways essentially evaluating accuracy. So here's looking at the clustering component, and we notice similar trends. And here Hintra is not included because they don't have a clustering component to their algorithm. They just identify patient trees. But again, as the number of subtypes essentially increases, we see that our performance remains relatively high. We also then ran our algorithm on biological data. So here I'll present results from a breast cancer cohort. We also looked at a lung cancer cohort. So in this cohort, we had over 1,300 patients, and the number of trees per patient in the input really varied, with one patient having over 6,000 trees. We also restricted the data to mutations that were sufficiently reoccurring and we felt like we could find patterns from. So in this case, they occurred in at least 100 patients. And then we also, through our algorithm model selection subroutine, we identified essentially 8 clusters in this data set, which ranged from 55 to 400 patients in each. So relatively well balanced. And here we see essentially the model selection component, where there's after 8 clusters, there's sort of diminishing returns to increasing the number of clusters. And then in panel B here, essentially this is trying to show the stability in our clusters. So as we increased K, we noticed not just like completely new types of clustering, but really subdivisions of existing clusters for lower values of K. So in that case, we found kind of almost like a hierarchical clustering to our patients. And so here's an example of sort of the utility of our algorithm in practice. In a sort of simple case, we have a particular patient with 2 different input trees, and there's ambiguity in the relative ordering of 2 driver mutations. And essentially recap here notices, quote unquote, notices that then another patient had resolved the actual ordering of these 2 mutations, and this informs a consensus graph that we use that orders these 2 mutations, and this ordering is supported by 11 other patients in the cohort, and then ultimately that allows us to select output patient, the second tree for this patient, consistent with the other patients in the input data. And then we also show that basically Hintra had also been run on this data set. However, they had initially manually split patients into 4 different subtypes based on receptor status. And we show that we recover some similar results and essentially recover one of these receptor subtypes without noting it up front, and also find a similar pattern for this subtype. And so just in conclusion, I just want to highlight that this is, really an adaptable framework, I think. So this is a first step, but I believe, we can look further into changing different distance metrics, looking at different ways of thinking about consensus graphs. Do we need to think about consensus as a tree, or are there other type of graphical models that would better represent sort of similarity and shared history, and as well as other models, and it's available on GitHub. And with that, I'd like to thank my co-authors and open it up for questions.
Thank you, Sarah. So we have several questions. I don't know if we will have time to answer all of them. So the first one was by Peter. So instead of constructing a full consensus tree for stratification of patients, will it not be sufficient to mark for each patient which mutations are highly probable to be early events and which are late, and then stratify based on early events?
Yeah, so I think this is a question that we see with the founding mutations being at the trunk of the tree versus later mutations. In terms of sufficiency, I think that's sort of a question that needs to be tested. So using essentially phenotype data, can we find meaningful stratification into clusters? I think on the particular cohorts we were looking at, just stratifying patients based on essentially sort of these founding mutations versus later mutations, we weren't finding meaningful stratification in terms of prognosis or time to death or different things like that. That's not to say that that wouldn't be the case in other cohorts, and I think that's always like a really good baseline to be benchmarking, and considering your method against. But in our particular biological data sets, we weren't finding sort of like meaningful phenotype information by doing it that way.
Okay, thanks. So next question is by Salva Capella. He says, nice talk. Why are you restricted to 8 mutations for the breast cancer cohort? It seems an arbitrary number.
Sure. I mean, I guess I would agree in some sense that it is arbitrary. There's not like a hard science to how we restricted it, right? I think the intuition behind it was that there was a lot of mutations that just only occurred in a handful of patients. And so when we're thinking about patterns and looking for patterns, we wanted to find things that were sort of well supported by enough patients. But our algorithm would run on without the restriction. In fact, we ran it that way. So it's not an algorithmic limitation per se in running time or anything like that. It was more just, we weren't finding that essentially if a mutation isn't in enough patients, it won't have sort of enough support to ever be included in the consensus tree, because the consensus trees, in order for something to be there, like mathematically, need to be occurring in a sufficient number of the patients in that subtype or that cluster. So they sort of all ended up falling out in a sense anyways, because they would never be supported enough. But again, that's a function of our optimization problem. And so potentially if you think about your optimization problem in a different way, maybe there's a better and more meaningful way to incorporate those less frequent mutations into your analysis.
Okay, thanks. So maybe we have just a few seconds for the last questions by David Plaza. So he says, you plan for the future to include the structural variants, e.g. from PacBio data?
Yeah, so I think that is something we're looking and trying to include more into the future, moving away from just looking at these single nucleotides or somatic mutations. And so that is a direction I think that will grow in conjunction with trying to do that better just on phylogenetic tree estimation as well. I think that's definitely an area of interest we're looking into.
Okay, so thank you, Sarah. I think with this, I can thank all the speakers also for keeping up with the time, and all the attendees for the interesting questions. And we close the session now, and I hope you enjoy the rest of ECCB. Thank you.